All right, we're starting. Thank you. Okay. All right, so today we are welcoming Louis-Marie Bobay, who is a recent hire at NC State. He started this semester, so first, like right off the bat here. So Louis-Marie got his license in biology and chemistry, which is equal to a Bachelor's of Science degree at the École Normale Supérieure of Lyon, France, and then went on to finish a master's degree at the same institution. Then moved to get a PhD at the Pasteur Institute, which he received in 2014 in genomes and genomics and genetics. He then moved to the US to take a postdoc position at the University of Texas at Austin. And in 2018, moved to UNC Greensboro in a faculty position. During that time, he was incredibly productive and received several grants, including an NIH and R01. At some point, we opened up several positions as part of our faculty of excellence, the cluster hire positions in the bioinformatics group. We had several vacancies, so we had 2 positions that came open. And so Louis-Marie came to join us, again, starting now. So he's in biological sciences here at NC State, and I'm going to let him tell you about his work.
Great. Thank you so much for the introduction and for the invitation. So like Dalia mentioned, I used to be in that room not so long ago for my interview talk. So if you were in that same room, you might see that some of the content is similar, but the good news is that about half of the presentation is new stuff. So you shouldn't be completely bored. So I'm going to talk about recombination in bacteria today. So we have several projects going on in the lab that revolve around speciation and population evolution, recombination, but also adaptation, evolution of gene diversity in bacteria and metagenomics. But today I'm going to talk only about the two main, the two main topics today are going to be about speciation and population structure in bacteria, and about homologous recombination in bacteria. So if you're like me, that your background revolves around evolutionary biology, you will think about species as being something that is capable of gene flow, or at least one of the offshoot of the biological species concept. One of the biggest issues with biological species concept is that it's not applicable to different species when there is no sexual reproduction. So one of the big questions that has been going on for years is, what do we do about microbes? Microbes do not reproduce. They are therefore not applicable. You cannot apply this kind of definition to them. And how do we define species? Well, you must first ask, do we even need to define species in bacteria? And actually, the answer is yes, because if you look at the diversity of bacteria, you can actually realize that they tend to form clusters, that the distribution of genome is not completely random. It's not a continuum. It's not something you would expect if there was no forces maintaining some sort of cohesion. So we do, when we compare bacterial genomes, we do see this clustering happening, and that strongly suggests that some forces are at play to maintain some sort of clusters that we want to call species. So what are we doing right now to define species in bacteria and microbes in general? Well, we typically use arbitrary thresholds. So it has been defined empirically that a good number to define bacterial species is if they are about 95% identical or more, they are considered to be part of the same species. Okay? It is simply based on these distributions of genome similarity, and that looks like after 95%, you tend to have a gap. More or less. So essentially what you do, and it's not very satisfying, obviously, but you take one reference strain for a species. And then if you want to classify some new genomes that have been sequenced, if they're 95% identical or more, they're part of the same species. If not, they're different species. So the problem is that there's a clear lack of foundation, theoretical foundation for why that is. Why do we want to define species this way? And is it even relevant to define species this way in bacteria? So I've told you the biological species concept is not applicable to bacteria. However, even though bacteria are not sexual organisms, they do engage in gene flow. And more and more people are starting to think that, well, maybe gene flow is actually significant enough in bacteria that it could actually be used as a cohesive force as it is for sexual organisms to maintain species. So even though gene flow is happening in bacteria, it is actually quite different than from sexual organisms. If you're typically used to sexual organisms, most of the recombination occur through meiosis during crossovers, where entire pieces of chromosomes are swapped in rearranged with each other. However, in bacteria, but it also happens in eukaryotes as well, this process of gene flow takes the form of gene conversion, where small pieces of DNA, typically a kb, 1 kb or less, are exchanged between individuals. So when I'm going to talk about gene flow in bacteria, I'm going to talk only about these small events of gene conversion that only involve small pieces of DNA. Another thing I like to clarify is you hear a lot of horizontal gene transfer in bacteria, and there are lots of papers that have been showing that horizontal gene transfer is common and is common between distant lineages. However, we're also not talking about the same process here. When we talk about gene flow, I'm talking only about homologous recombination, where small pieces of DNA sequences recombine with each other. So this only happens if the DNA strands, the sequences are actually very closely related. When in HGT, you typically have brand new genes, brand new sequences that are introduced into a new genome. And these processes of HGT don't actually require homology very frequently. So some of the questions we are trying to address in this work was to understand how many bacteria actually do engage in gene flow, or at least in a significant way. And if so, can we define bacterial species based on gene flow? And also, eventually, we would love to understand is what is causing these interruptions of gene flow between bacterial species, and what may be like impacting the structure of bacterial populations. So we use the big data approach to answer this question, where we analyze essentially any reference bacterial species that we could find, meaning that whatever is called a species on NCBI, we started with that. And then we tried to redefine that. So essentially, we analyzed over 30,000 genomes of bacteria. We defined the core genome, so the set of genes that are shared between all the members of that species. And then we use essentially a shotgun approach where we analyze the signal of homoplasy in this genome. So homoplasy is essentially an allele that is not congruent with the topology of a tree. So what we do is that we look at every polymorphic site in the core genome of bacteria, so typically over thousands and thousands of sites, and we simply count how many of those are homoplasic versus those that are not homoplasic. And we define this simple ratio H over M, which essentially is the number of homoplasic sites versus non-homoplasic sites in a genome, in a species, sorry. And this actually gives us a good idea of how much recombination is going on, because these pattern of homoplasies are thought to be mostly introduced by recombination events. So the first thing we want to do is to identify which species in our data set are clonal, which ones do not recombine at all. So I've told you this signature of homoplasies are typically the signal that shows you that recombination has happened, but it's not completely true. In some cases, these homoplasies can be the result of conversion mutations. So independently, the same mutation happened at the same site and give the same allele. And that becomes increasingly probable as mutations accumulate over time. So what we wanted to do first is to try to estimate how many species in our data set have homoplasies that are only due to commercial mutations instead of recombination events. So what we do is that we use the simulator based on the species tree of each species, and then we simulate the evolution of sequences along that tree by conserving all the parameters that we could infer from that species, the level of polymorphisms, the GC content, and all of that. And then the idea is that if in the reference species, in the true species, homoplasies are essentially a mix of are results of both recombination events and gene conversion events, conversion mutations events. And in our simulated species, we did not introduce any recombination. So, which means that in this case, we know that the number of polymorphisms that are there, the number of homoplasies actually, are only the results of conversion mutations and not recombination. So what it tells us is that if we simulate these sequences and that these two ratios are the same, then it would tell us that actually the totality or the near totality of homoplasies in that species are expected to be the result of conversion mutations rather than recombination. So that's the results of this first analysis that has been, this entire analysis, by the way, has been conducted by Awa Diop in my lab, who's been a postdoc for several years in my lab. And as you can see, this line here in orange represents what you would expect if all the species were actually clonal, where all the homoplaces were due simply to conversion mutations. But as we actually see is that the vast majority of species have a clear excess of homoplaces compared to what would be expected just by conversion mutations. So based on that analysis, we expected that somewhere around 10% of the species of bacteria that we analyzed could be considered as purely clonal, so it did not recombine at all. So we still have like, the vast majority of species seems to show a signal of gene flow. We're going to confirm this result with a completely different approach. So another way to capture the signal of recombination is to use to look at the linkage disequilibrium, or LD. So simply put, linkage is when two sites are close to each other. We expect them to be all associated together a lot because they would be not by chance, they would be very unlikely that a recombination event would split them, right? So what typically you can do is look at two different sites in the genome, and as the sites are further apart, it becomes more and more likely that a recombination event will dislink them, right? So what we can do along the core genome of bacteria is draw this function here that is looking at R square, so the correlation of association of loci relative to distance across the genome. And when there is a recombination going on, you typically see this kind of pattern where R square decreases as you look at sites that are further and further away in the genome. And those are two examples that we obtained. For instance, here we have a case of chlamydia abortus where there is no correlation at all between the linkage pattern and the distance between sites. And here we have a case of a species where you have a clear signal of recombination, where you can see that linkage decreases as you look at sites that are further and further away. Okay. We have a ton of data, so we can have like very robust signal like this because we have thousands and thousands of pairs of sites along the genomes. And that's actually a cumulative representation of all these graphs that we conducted. So, like I said, if we expect recombination, we expect the graph to look like this one on the right, so meaning a negative correlation, a significant correlation. And as you can see, I mean, actually, what you cannot really see is in green here, the vast majority of the species we looked at have a clear significant correlation, meaning that there's a clear decay of linkage. And for a handful of species in orange here, we actually have no statistical association between distances and the decay of linkage. So based on that analysis, we can see that about 6% of bacteria in our data set do not show any signs of gene flow based on linkage. So if we look at these two analyses together, we actually see that if we take the two analyses together or separately, we have an estimate that somewhere around 2% to 13% of all bacteria are truly clonal, meaning that somewhere at least 86% of the species that we analyzed seem to be showing some clear signal of gene flow. So what we want to do next is then to try to redefine species borders based on gene flow for those that actually seem to be recombining. So we started applying an approach that I started doing my postdoc because it is actually sometimes it's not as trivial to identify some specific genomes that are giving an abnormal signal that actually decrease the patterns of homoplaces. So what we actually do for this kind of analysis is a massive resampling analysis. So if we start with a population of different genomes and we suspect that they are the same species, but if in this case, one of the genome is part of the different species and does not engage in gene flow with everybody else. So what we can simply do is start sampling the genomes, take, for instance, 100 times 4 strains, and then start by taking, and then increase the number of strains little by little. And typically when there is one genome that is not recombinant with the other ones, we see this type of patterns. As the sample size increases, at some point, we see a drop in the signal, okay? And that simply reflects that as you increase the sample size, you'll be more and more likely to include this particular genome that is like shifting the signal. So what we can do then is identify these genomes and redraw these graphs, and we can actually see a much more homogeneous level of homoplaces in the population. So we processed all the non-clonal species that we had defined. So we started with about 200 species. And what we did then was to compare all the related species around them. So any species that was somewhere at the related to the genus level, and that was about 2,400 species. And so we started by each of these species, the reference species, so the one we started with. We first check for the presence of genomes that may be not recombining with the rest of the population, in which case they were excluded from the species. Then we looked at all the other related species, the candidate ones, and we tested them individually for patterns of gene flow going on with the other strains of the reference species. And essentially, depending on what this signal will give us, we would actually either reclassify them as part of the same species or keep them into separate species. Okay, so that analysis actually took the supercomputer quite busy for a couple of years. But every time we actually calculate different metrics, what we call the ratio of homoplasy, H over M and ref, which actually represents the level of homoplasy just in the reference species. Then we do that by also recalculating that while including the candidate species as well. I've also told you that as we look at more and more divergent genomes, it becomes more and more likely that homoplasies are actually due to conversion mutations rather than gene flow. So to account for that, we also run simulations for each degrees of divergence that we observe in our data to estimate the amount of conversion mutations that are giving rise to homoplasies. So essentially, then we can use this metric that is the adjusted level of homoplasy, H over M norm here. And what it means is that essentially when it's close to 0, that means there is no gene flow. And when it's closer to 1, that means that we do see a lot of gene flow going on in the two, pretty much the same level as you see within the reference population. That's what the distribution looks like. As you can see, for the vast majority of pairs of species, we see no clear signal of gene flow since everything is very close to 0. But actually, there's so many pairs here that we can actually not really see what's going on. But if you look at a different representation of that, you can see that individually, we see that there's actually quite a lot of pairs of species that are right there, where the levels of gene flow is actually the same whether or not you include a different species, which strongly suggests that those are still engaging in gene flow at the same level as the reference species. So based on this analysis, we estimate that around 8.5% of species actually that are classified into different species would be classified as the same species if we were to define them based on the patterns of gene flow. What does it look like compared to the similarity threshold that are typically used to classify species? Well, the one that would actually reclassify into the same species, yes, they tend to be closely related, but actually you don't see a clear, sharp delimitation at 95%. Typically, most of them are above 90%, but 95% doesn't seem to be the magic number that can delineate species. And also conversely, you can see that the ones that we did not reclassify together, sometimes some of them can be above 95% identity on that genome, but actually do not necessarily engage in gene flow. We also want to show you some examples of what we redefined. For instance, if you look at E. coli, so we built the tree with all the relative of E. coli. For E. coli, we only included 10 genomes to make the tree. And as we can see, like most of the relative of E. coli are considered different species, except Shigella. So Shigella, I don't know if you're familiar with that, but actually Shigella is not even a different genus, not even a different species of E. coli. It's actually, there are actually different strains of E. coli that just gain a plasmid and then can become pathogens. And that's actually very consistent with every other criteria to classify species. We know that based on identity, based on many other things, Shigella is actually part of E. coli. So we did find that as well. We also looked at, got some more interesting results. For instance, in Burkholderia stenocepacia, where in this case, you have the focal species in dark blue. And we see that actually a lot of related species actually engage in gene flow at the same level. And that's why we suggest that they are part of the same biological species, even though they are actually in this case more divergent. Some are only like around 90% identical. But it's funny because also microbiologists often refer to this clade as the Burkholderia Cenocepacia complex, that they are often treated as one anyway. So this I thought it was interesting. So I've shown you that, yeah, we can potentially reclassify species based on the presence or absence of gene flow. But I guess as you could suspect from the graph I've shown you, there is not a clear necessarily barrier of gene flow, right? There's not a clear point where there is gene flow or not, and it's more like a continuum. So we wanted to look at to what extent gene flow is still maintained between species that are actually considered different species based on every criteria, including our criteria. So we call that introgression because just like for animals and plants, you can see that sometimes species are not fully separated and that gene flow still occurs. There are many, many examples of that. For instance, between the grizzly bear and the polar bear, also between humans, within modern humans, and I've got some introgressed DNA from Neanderthals and Denisovans. But actually, a very interesting paper came out several years ago in bacteria, where it has been reported that there's been some massive amount of introgression between two species of Campylobacter. And again, you think, well, DNA transfers is not crazy in bacteria, but in this case, it's very, very interesting because we're not talking about accessory genes that are exchanged and gained. We're talking about the backbone, the genomic backbone, where about 15% of the entire housekeeping genes have been swapped by on the entire sequence. So it's a massive amount of genes that have been exchanging alleles. And also, what's important here is that these two species are only 85% identical. So it's a massive amount of DNA that is exchanged for two species that are quite divergent. So we're going to see if there were other cases like that. So in this case, again, we wanted to detect these cases of gene flow, but this time between species. We didn't want to use an approach running on homoplasy because, like I mentioned, as you look at more divergent genomes, the levels of homoplasy just simply accumulate just by conversion mutations, and things become less and less accurate. So what we did instead, we used a scanning window along all the pairs of species we had. And the idea was simply to try to find some pieces of DNA in one species that was much more closely related to another species, in this case, species A, than any other genome within that species. And those would be indicative of relatively recent gains of DNA from a different species. Then for each pair of species, we actually computed what portion of the entire core genome is actually the result of introgression. And we calculate that as the percent of the entire core genome. So that's the result we got for all the pairs of species we got. So we found that about half of all the species pairs we compared show at least 1% of their core genome being recombined with one another. So that suggests that introgression is probably quite frequent in bacteria, but it's typically at pretty low amount. And there were very few species that are above 15% of introgressed DNA. But still, it suggests that it's relatively common. What we thought was very interesting is that we found that these levels of introgression were not independent at all of the divergence of genomes. If you look at this graph, you can see that typically the species that are most closely related tend to exchange most DNA. And as you go down in divergence, you actually exchange less and less DNA. Again, that was expected, but what I think was much more interesting in this case was mostly the shape of the distribution, where we have this sort of exponential relationship between genome identity and the levels of introgression. So that led us to hypothesize what was maybe going on here. What's going on and why do we see this exponential relationship between sequence identity and introgression? Well, in the 80s and 90s, people started to publish some papers about the mechanistics of recombination in bacteria and how does that work. And people have essentially identified that to successfully recombine, you need to find identical stretches of DNA to be able to initiate recombination. Okay, so that means is that even though in theory you can recombine pieces that are only 95% identical, well, what really matters is if they are or not strictly identical over a small fragment. And the size of these fragments, called MEPs for minimal efficient processing segments, has been shown to vary across species. Okay, it's actually been only defined for 4 different species for what I could find. But depending on the species, the size of these fragments that need to be identical for a combination to be initiated was somewhere between 26 and 90 nucleotides. And what it means is actually might have some pretty important implication for this, because here simulations are rerun, and essentially based only on the size of these MEPs, what we can do is simulate what we expect our combination to look like across genomic divergence. So here we have at 100%, we have 2 identical genomes. Of course, we'll find a ton of segments of DNA that are identical. So there will be essentially no problem to recombine. But what's interesting is as you look at more and more divergent genomes, you see this also exponential relationship where it becomes less and less likely to find small identical stretches of DNA. And that, of course, changes at a rate that varies across the size of the MEPs. But and that's again our results for the introgression level, for the introgression events that are the most recent, so the ones that are 100% identical. And so what it suggests to us is that this is maybe the dynamic that is causing this sharp decline in recombination rate between these species. Also, I want to show you this graph in the 90s that's been shown experimentally that it is true. Fred Cohen and colleagues did this analysis where they looked at the probability of 2 sequences to recombine and at different levels of divergence. Okay. It doesn't look like this because it's a log scale y-axis, unfortunately, but it's actually, they also observed an exponential relationship between the two. So as sequences divergent, become exponentially less likely to recombine. So what we think is going on is that after a certain level of divergence has been reached, it becomes mechanistically almost impossible for 2 bacteria to recombine, just because it will be impossible, almost impossible to find stretches of identical DNA that are at least 25 or 50 nucleotide long. And also what we think is very interesting is that if you look at the shape of this distribution, it actually matches quite well the levels of sequence divergence that we observe where most species tend to be completely completely stop exchanging DNA. So I think this idea that 95% identity is a good threshold overall to define species, we have shown it is not accurate, but what we think is going on is that it's just simply based on this simple expected patterns of recombination based on the size of MEPs and based on the divergence level of genomes. So I'm also not trying to say that just by simple genomic divergence, this is what is causing speciation and interruption of gene flow in bacteria, because we think that there's other more important forces that drive this divergence in the first place. But what we're thinking is that those are the mechanisms that make it so that once 2 species are speciated, it becomes even more complicated for them to exchange DNA. So in conclusion for that part, we have been showing in this study that few bacteria are truly asexual. And actually, it's in line with previous work that I've also showing that, well, actually, it's actually harder to find bacteria that don't recombine at all than to find bacteria that recombine. We can see that the genome 1 threshold that are typically used to define species of bacteria are not accurate at defining species if we compare that to our methods by looking at patterns of gene flow. And also, we believe that sequence divergence acts as a very important recombination barrier that strongly reinforces speciation in bacteria. All right. Now I'm going to show you some very recent results, and I'm excited to present for the first time. And it's also about recombination. So I've told you that people tend to think of bacteria as being clonal, even though there's some clear evidence of recombination going on in bacteria. There are actually some studies that have suggested that bacteria, some species of bacteria, can recombine at such high level they can be considered quasi-sexual organisms. And it's been actually quite an interesting problem because it's been very not clear to what level of bacteria recombine. There's been quite a few papers on bacteria for like decades that have been shown that recombination is there. But how much does it actually recombine? And that's a very important question, because what processes best describe evolution in bacteria? Okay, when you develop models for bacterial evolution, should you more follow a clonal model or more like a similar models that you would use in animals and plants? Okay, so that's actually a big problem, because lots of methods are being applied to bacteria just as if they were completely clonal. Other methods are applied to bacteria as if they were completely sexual. But this is not clear which is better, or if both are completely unadapted to these organisms. So apologize for this pretty scary table right here. But there has been actually two big studies that have looked at the recombination rates across all bacterial species. And that's probably the biggest one in 2009. Both indeed looked at recombination rates across all the bacteria they could find data of. Note that it was on MLST data, so only these marker genes, so only like typically 7 marker genes per species. Nowadays, we do that with complete genomes. But what was interesting in this study, and I'll explain that later, is that the recombination rate here varies essentially between 0 to 63, which is quite interesting, and because it actually spans all values in the middle as well. So meaning that bacteria can be either completely clonal or recombine at extremely high rate. And so we seem to have such a diversity of propensity to recombine bacteria. So a few words about how this is calculated. Most methods attempt to calculate what is called an effective rate of recombination. Effective in the sense that it only matters if alleles are exchanged. If you exchange pieces of DNA, but the two pieces are highly identical or completely identical, it doesn't matter because it doesn't change anything. So those rates are defined essentially as the number of recombined alleles relative to the number of mutations that have been accumulated. So essentially, if you look at a tree to look at the evolution of the population here, you have several mutations accumulating in each branch, and some of these alleles are exchanged. And all over M essentially describes the ratio of how many alleles are exchanged by recombination rather than introduced by mutations. Okay, so they have quite a lot of methods out there. Some rely on the detection of homoplazy, some rely on the patterns of linkage equilibrium. So there's quite diversity. And even though there's a lot of methods, there's been a lot of issues with that. The main issue is that the estimates are highly inconsistent across methods and across studies. And it's not really clear why that is. Okay, so people have argued that, well, we never get the same estimates of recombination rate across species because the methods are different. And it's true, that's often the case, but also the data sets are different, So if within a species, there is a lot of structure and that some population recombine way more than others, depending on what your sample is, you might get very different estimates, even though the methods are the same. So we don't really know what's causing that. And another issue is that most methods rely on the direct signal of recombination. That can be an issue because many recombination events don't leave any trace. Like if you have a single allele in a single strain that is exchanged to somebody else, it doesn't necessarily leave any pattern of linkage, any pattern of homoplazy. And to finish on this issue, the other issue is that most methods typically rely on one specific signal of recombination, typically either homoplazies or linkage. So what we want to do here is to use a different approach. We will integrate different signals of recombination and do something a bit different. So we use an ABC approach for that, so for approximate Bayesian computation. So if you're not familiar with that, it's a little bit like machine learning, but instead of training the data on real data sets, you would train them on simulations, for instance. But the idea is that for each species, we have a core genome here, and we want to know what the recombination rate of that population is, that species is. The way we do it is to build a tree from that thing. And also estimate a bunch of other parameters, including composition, transition ratio, the level of polymorphisms. And the goal is to simulate using that tree a bunch of other data sets that would be recombined at different levels. So we know in this case what recombination is because that's us who introduce more and more recombination. And the idea is that once we have all these simulated data sets, is try to find the one that shows the signature of recombination that match the real data the most accurately. So that's an example. By the way, all this work has been led by Elise Torrance in my lab. She's a grad student. She will be defending quite soon, actually. But she has been also running in that for years and making the supercomputer very busy. But this is an example of how that works, for instance. So we would simulate a data set 360,000 times. Typically, now we would do it 500,000 times. And we have simulated these sequences with different levels of recombination rates. And then after that, we look at different summary statistics that we know are associated with recombination rate. The level of home places, the level of polymorphisms, and the function of decay of LD. And the idea is that from all the simulation, this is in gray, you see the different recombination rate of all our prior distribution, so all the different simulation. And at the end, our ABC approach will essentially find the simulations that most closely match the real data set based on these 3 summary statistics. And if we did a good job, typically it converges towards one value that is clearly different from the peak of the prior distribution. So that's, I'm not going to enter into the detail here, but that's essentially the results of several years of supercomputing running for nonstop. But I'm not going to discuss that in detail right now, but all I wanted to show you is that we again obtained a wide variety of recombination rate across species. Okay, we were able to analyze over 160 species. And what we can see is that some species are clearly like no recombination at all, and some are recombined at pretty high rates. Again, we've got pretty much all the values in between, with no clear patterns of anything going on. Because I've told you there's so many issues with the validation of methods and methods being inconsistent, we wanted to validate our approach as thoroughly as we could. We first checked whether or not we could retrieve the summary statistics from our simulation. In other words, if we did a good job, we should find a simulation that have summary statistics that closely match the real data. And for the most part, we did. There were some that were not very accurate, so we had some estimates that are a bit more questionable. But the summary statistic that was the most problematic was the decay of LD, and that's because unfortunately, that is very, very hard to get something accurate when there's very little levels of polymorphisms, which is the case for some species. We also validated that against a bunch of simulations. Unfortunately, this is not the figure I wanted to show you, but I couldn't find the figure I wanted to show you. But this is actually showing that we were able to accurately identify the recombination rate based on simulations, right? So we could discard a bunch of the simulations, not use them to predict anything, and actually identify what values were simulated for the simulations. We did identify, however, several cases where we could not accurately identify recombination rates for the simulation. And that was actually typically, again, when polymorphisms were very low, when there was very little signal. In this case, we could not accurately identify that. However, when that was the case, there was usually when there were actually very little recombination going on, and actually we knew that was actually happening. We also wanted this time to see, well, because it is so inconsistent across studies, and maybe it is due to different sampling, what happens if we resample our data set? So what we did is that we removed 10 genomes for each data set, so we represent about 10 to 40% of the size of each data set, and see if we are capable of retrieving the same recombination rate or not. And for the most part, the answer is yes, except for a few exceptions, we were able to accurately identify the same recombination rate by even though we subsample the data sets. Another thing we did was to do another resampling analysis. This time we used E. coli to do this analysis. Why E. coli? E. coli because it's actually very known bacterial structure in that species. So it's been defined for decades, and every time new genomes have been sequenced and added to the story, we still find the same population structure in E. coli. So we know that E. coli is divided into several main groups, A, B1, B2, D, and E. And what we did is, starting from a completely different data set, it's actually an old data set that I built several years ago. I wanted to see if we could actually re-estimate the same recombination rate by doing some subsamplings. So in this study, with our new estimate, we have a value of recombination rate that is somewhere around 3.5. And what we did using a completely different core genome built with different strains, we actually resampled them across the different clades. And that's the value we obtain, that are not that different from the ones built on a new data set. What was interesting to us is that despite the fact that there are some clear structural population in E. coli, we did not really identify any big difference in our recombination rate across the different phylogroups. Some phylogroups seem to be a little more consistent with each other, but there was not, some, on the other hand, were more divergent than some of them. But essentially, we're actually surprised to see that there was no strong differences across the default clades. So, that really suggests that estimates of recombination rate seem to be very robust to genomic sampling. Of course, we only did that in E. coli, so it would be interesting to repeat that using other species. Also, we compared the results to the two big studies that have been done before on recombination rate. And interestingly, our results are also very different. So these two studies estimate recombination rates across a bunch of bacterial species. And they were very inconsistent with each other. And now we are also inconsistent with both of them. There is a slight significant correlation with the first method here that was it. But again, this first study was used on using MNST data, so a much smaller set of genes. And what we also did was to look at how recombination rate evolved in bacteria. So we built a tree for all the species we had in our data set. And we used the maximum likelihood approach to reconstruct the rates of evolution at every node of the tree. And based on previous studies, I didn't expect they would see any clear pattern, but we did observe some interesting things. We observed that actually, it seems that in some genera, there is actually quite conserved recombination rate. There's not a completely chaotic variation. I'll show that in another figure, but essentially we saw that in Bifidobacterium, we have consistently higher recombination rates. We also found that overall, Streptococcus is one of the highest recombination rates, and that was quite different. But that's a representation of all the genera we analyzed. And essentially, like several of them were found to be significantly different. We found that Bifido and Streptococcus were recombining at higher rates overall than everybody else, and in a consistent manner. Also, two other genera, Staphylococcus and Chlamydia, were also found to recombine at lower frequency. Another thing that is interesting about our method is that, as opposed to most methods which are estimated on effective recombination rate, so it's highly dependent on how many alleles are actually exchanged, we can have a more absolute estimate of recombination rate, because we don't only see what is exchanged if it's a polymorphic allele, we actually see the total number of nucleotides that are exchanged. So if we compare all values that we estimated that varies between 0 and 30, we actually see that in terms of total number of nucleotides that are exchanged, it varies somewhere between 0 and 10,000. Okay, so that's still relative to the mutation rate. And 10,000 for doesn't mean that you have a block of 10,000 exchanged at once. It could be multiple events of 1,000, for instance. But what we thought was interesting is that it looks like after a certain amount of recombination, typically when you start reaching 10,000, we actually become, it doesn't seem to really matter anymore for the effective recombination rate. So which suggests that we actually saturate after a certain level. So what we suspected, because as you can imagine, as recombination increases, the patterns become more and more chaotic. It becomes like events start to rewrite each other. And at some point, we don't expect to be able to identify all these events. So we expect that it should be saturating at some point. So, but based on this graph, what it suggests is that it's actually saturating quite early at all RAM equals 5, which probably means that you should not really trust anything that is above 5. We also looked at a bunch of other, when I say we, was Alice, we did a ton of work. She looked at a ton of other features that were correlating with recombination rates. And several of for the most part, we didn't find any clear correlations. One that we found was that as you increase the number of genomes, you typically have a higher recombination rate. And we believe it's simply because you are more likely to have a better representation of the species. And then if you are undersampling a species, you most likely underestimate recombination rate. But what's interesting is that as opposed to previous studies, we didn't find any pattern of recombination rate and pathogenicity. There have been some being claim made that recombinant bacteria, the pathogens are typically recombining more. We don't find any evidence for that. Also, there has been some claim that maybe GC content in bacteria is driven by recombination. Well, that might be true within a genome, but at the species level, it doesn't seem to have any trend going on. We also looked at a bunch of other features, like between whether the bacteria are heterotroph, autotroph, aerobe, anaerobe, a bunch of other things. Only one feature was clearly significant, was the lifestyle of bacteria. And for the bacteria we could qualify as free living, they were clearly different, mostly from obligate intracellular. So it's not a big surprise, but clearly if you are living if you are an obligate intracellular species, you clearly don't recombine much. All right. So, all right, to conclude on this part, this is the largest analysis of recombination rate that has been done so far. Also, it has been not done in a while. And so even though we can say that recombination rate vary a lot across species, we still have a lot of differences with other methods. But at least I believe that we tested our method more thoroughly than maybe has been done in the past. Also, we have found that it's quite interestingly, recombination rate seems to be a rather slow evolving trait. It's not the case in every lineage, but it seems to be rather conserved in some lineages. Also, I want to give you a short overview of all the other projects going on in the lab, because besides that, we're also working on speciation of host-associated bacteria. Notably, Carrie in the lab, she's looking at microbiome in honeybees and how the speciation of honeybees and bumblebees, for instance, do impact the speciation patterns, but also the gene flow pattern of species in the bee microbiome. We're also doing some work with Shiva Thapa on the evolution of strain dynamic and strain community. And both Shiva and Carrie are co-advised with the lab of Casey Raymond, who works on honeybee microbiome. We're also working on some metagenomic analysis. We're right now developing a tool to resolve the genotypes of bacterial strains from shotgun metagenomic data. So we can hopefully follow what's happening in more complicated data sets. And that's a postdoc in my lab, who is Hazem Sharaf, who is conducting this analysis. We're also looking at evolution of genome rearrangements in bacteria. And that is conducted by Carolina Martinez in my lab. And Awa, that you saw the first results of earlier, is also right now working on introgression in bacteria and trying to quantify how much introgression is going on in bacteria. All right, well, I would like to thank everybody in my lab. And thank you for coming and for listening to me.